Welcome to the second part of this talk. Here we will discuss what a cell looks like. So we probably all have some idea of what a cell looks like, and it might be heavily influenced by textbook images or by images such as these. I think I took this image from Wikipedia and it tries to show you what a cell looks like. But there's a couple of things that are wrong with this image, I would say. So why don't you take a couple of seconds to think about things that could be wrong here, and then we will discuss them. So one of the things that is not entirely correct is the amount of organelles that you see. For instance, you see only a handful, 3 mitochondria. Their shape might be off. Another thing is that this Golgi apparatus just looks like the endoplasmic reticulum. And then another important point is that a lot of these spaces are present in this cell. So empty spaces where no molecules or organelles are located. Now let's try and see whether we can get a more realistic view of a cell. Now I like these kind of graphs, these drawings really a lot. They're made by J.Goedhart, an artist that tries to make realistic drawings of cells and other kind of biomolecular structures. So what you see here is part of a cell. This complete gray area is the nucleus. Here's a part that is highlighted to show endo and exocytosis that's enlarged over here. And on the next slide we can also see a zoomed in part of a cell. So what you see in this image is that the cell is completely packed with all kinds of macromolecular structures and molecules. So for instance, here you can see a ribosome. We see a membrane structure with transmembrane proteins. Right here we see a proteasome and we see filaments running through the cell. And what you should appreciate from this image is that the cell is literally packed with all kinds of molecules and structures. Now these drawings are nice, but you can also directly observe that. That is shown in this example where we have used GFP markers with a specific targeting sequence to, for instance, highlight peroxisomes or mitochondria. Here's the ER, and you can see the cell is completely packed with this ER. And here we have actin labeled. And in any of these images, you see that there's lots of these structures around in the cells. Now there's another thing that you cannot appreciate from textbooks, and that's of course dynamics. So to show you some cellular dynamics, this is a movie of HeLa cells, so cancerous cells, that is imaged overnight. And what you see in this movie is that the cells are dividing and actively moving as well. So it gives you a much better feel for the dynamics of these cells. Now also within cells, molecules are moving. So this is an example of a DNA repair protein that was tagged with GFP, and it's present in the nucleus to repair DNA damage. Now, from a static picture, you cannot appreciate the dynamics. So we did an experiment that's known as FRUP, fluorescence recovery after photobleaching, in which we photodestruct a couple of those molecules to give rise to this black stripe. And then we just take images over time, and what you will see is that those molecules exchange. So after a while, this black strip where the molecules were initially are photodestructed is completely filled again with fluorescent molecules, showing that those molecules are indeed moving throughout the nucleus. And you can quantify that and determine half-lives, and you will see that the half-life of this movement is around a couple of seconds. Here's another example of such a FRUP experiment. In this case, it's a plant cell. In this case, tubuli, microtubules are labeled. And from this static picture, it looks like the molecules are just fixed on the filaments. But now when we bleach away a portion in this white rectangle, what you will see, they disappear here, but after a while they recover again. Again showing that those molecules are moving throughout the cell. So this FRAP technology is a very nice way of looking at mobility of molecules in cells. Now there's another experiment that I really like and that nicely shows the mobility, and that is shown over here. So in this case you see a single cell in which we have introduced a GFP tagged to a binding domain, and this binding domain can label plasma membranes when calcium levels are elevated. So in a resting cell, calcium levels are low and the domain is distributed everywhere. Now we added to these cells something to stimulate them to release calcium, and what you will see when the calcium levels are increased, that this membrane binding domain moves to the plasma membrane. So I run the movie, and what you will see at specific times, you see a depletion of the cytoplasm and an increase at the plasma membrane. And this shows you that this molecule that is floating around in the cytoplasm is nicely recruited to the plasma membrane and binding there. And you can appreciate that this is a very transient and rapid process. And this is based simply on diffusion of molecules. And again, it shows you that those molecules are moving throughout the cell with quite amazing speeds. Now, all these microscopy experiments give you a slightly different view of a cell. So, to summarize those, cells are dynamically organized. You could indeed say that they are very well organized piles of molecules. They are full of biomolecules, complexes, organelles. They're completely packed with all these kind of structures and molecules. And even so, the molecules within a cell, proteins, they can really move fast. So they can move from one side to the other side of a cell within seconds. So cells, you should also appreciate, are highly dynamic. And I really want to urge you to forget this textbook image. And hopefully you will get a chance to observe cells and learn to know what they really look like. Now, the rest of the talk will mainly deal with signal transduction. And we will be using microscopy as a tool to study the signal transduction. And so I would like to specify a couple of points why we really are why we are really fond of microscopy to study those processes. Now we can study with microscopy these processes with very rapid speeds, so at video rate imaging, so at timescales down to the milliseconds. We can also observe where processes take place within a cell, and those images we can quantify. So we actually obtain quantitative data from these microscopy images. It should be clear by now, also from the movies, that we can really watch the molecules in action. And we get a feel for heterogeneity, because not all cells may respond equally. Some cells may respond different than other cells, even though they're genetically identical. And we get a feel for this heterogeneity when we do live cell imaging. And we can watch all those molecules in their natural environment, which of course is a living cell. And hopefully by watching these movies, doing perturbations, we can learn something about how those molecules act in living cells. So that's the end of this part, and then we will continue with the role of G protein coupled receptors in signals transduction. And again, I will explain how we can use microscopy to learn more about this process.